Today, we will be discussing about the oxidative phosphorylation, an essential process in cellular respiration that generates ATP. Let's begin by defining the process and identifying the key structures involved. Oxidative phosphorylation is the ATP production process using energy from electron transport chain in the inner mitochondrial membrane. High-energy molecule NADH and FADH2 from glycolysis and Krebs cycle transfer electron through electron transport chain complexes such as NADH dehydrogenase complex 1, succinate dehydrogenase complex 2, cytochrome C reductase complex 3, cytochrome C oxidase complex 4 via carrier ubiquinone coenzyme Q, and cytochrome C driving proton pumping. This creates a proton gradient that power ATP synthase to generate ATP during chemotherapy. Oxygen will act as the final electron receptor, forming water and completing the oxidative phosphorylation process. Would you like to dive deeper into this process? If so, let's explore the detail.
The process is known as oxidative phosphorylation. It occurs in the inner membrane of mitochondria. So come over and take a closer look on how oxidative phosphorylation really works.
Here, NADH transfers the electrons to NADH dehydrogenase complex 1. NADH is then oxidized to NAD plus 2H plus 2E. Electrons in complex 2 from Krebs cycle are then transferred to coenzyme Q. Also, at the same time, FADH2 is oxidized to FAD plus 2H plus 2E. From coenzyme Q, electrons are then transferred to a long series of electron transport chains, which consists of cytochrome C reductase, cytochrome C, and cytochrome C oxidase. Electrons are then transferred to O2, which acts as a final electron acceptor. O2 then combines with 2 electrons and 2H, forming water molecule.
Next, after the electron is transferred, energy is released. Then, the three of this complex, which is complex 1, NADH dehydrogenase, complex 3, cytochrome C reductase, and complex 4, cytochrome C oxidase, use this energy to pump out the hydrogen ions, or should be known as protons, out from the matrix of mitochondria into the intermembrane space. And so does the other. Then, this would create proton gradient across the membrane. This is because the hydrogen ions are high concentration in the intermembrane space and low concentration in the matrix of mitochondria. After the proton gradient occurs, it will create high concentration of H+ in the intermembrane space of mitochondria. So, it will undergo chemiosmosis. Chemiosmosis is a movement of hydrogen ion from high to low concentration of H+ through ATP synthase to catalyze the synthesis of ATP from ADP and phosphate group. H+ gradient causes H+ to diffuse back into the mitochondrial of matrix down the concentration gradient through ATP synthase. For each glucose molecule that enters the cellular respiration, chemiosmosis will produce 24 ATP molecules through oxidative phosphorylation in ATP cells. Next, the ATP synthase, if activated, catalyzes the formation of ATP from ADP plus phosphate groups. Let's find out how many ATP will be produced during the oxidative phosphorylation. So, one molecule of NADH will produce 3 ATP, while one molecule of FADH2 will produce 2 ATP. So, 10 molecules of NADH will produce 30 ATP, and 2 molecules of FADH will produce 4 ATP. In conclusion, during the oxidative phosphorylation, there was 34 ATP that will be produced.
Well, chemiosmosis is an essential life process for ATP production in living organisms. It occurs in mitochondria during cellular respiration and in chloroplasts during photosynthesis. The movement of hydrogen ions across the membranes creates an electrochemical gradient which drives ATP synthesis through ATP synthase. So, chemosmosis is a fundamental concept of biology and explains how cells generate and serve survival. Without chemosmosis, cells will not be able to produce energy for essential life processes. That's all from us. Thank you.